So I warmly welcome all of you to the day 2 session of this 1 week faculty development program. And now I'd like to invite the convener of this session, Dr. Chebrolu sir, onto the dais. Great privilege. I'd like to invite today's speaker, Dr. Hanumaiah sir, onto the dais. So now we are on the day 2 of this 1 week faculty development program on exploring potential of natural resources. So now I'd like to introduce the speaker of today's session, Sir Dr. Hanumaiah. Sir is the principal, Teachers College of Pharmacy, and he is acting as secretary of APTA AP State Branch. In addition to this, Sir has many achievements. Sir completed his B.Pharm and M.Pharms in Pharmacology from Andhra University. He completed his PhD in the year 2013 from Andhra University. And in total, Sir has 16 years of experience. And he guided around 60 B.Pharm projects and 30 M.Pharm projects. And he had a total number of 7 patents, out of which 3 are granted and 4 are in published status. And Sir had around 30 publications in various international and national journals. And he published around 2 book chapters and 1 book, that is textbook of pharmacology and phytochemistry. And Sir also acted as a convener, and he is the main chairman for various seminars organized by AICTE and APTA. And Sir also received a grant from GOC scheme of AICTE. And this, not only this, he also achieved many awards like Best Principal Award and from Association of Pharmacy Teachers APTA, and also from APP he received Best Appreciation Award. In addition to this, he had many awards. So with this brief introduction, I'd like to invite today's speaker to continue this session. Thank you, sir. First, thank you for accepting our invitation and coming far away to this place to give this session.
Happy to be part of this faculty development program. I thank Chowdavaram, who is my senior in pharmacy, Andhra University. I would like to convey my thanks to the principal, Dr. Vidyadhar and Chowdavaram, for providing me this opportunity and to have a part in this FDP program. So thank all the organizers and students. Today I would like to give you a brief review about the phytozoans. So I would like, my topic is a review on phytozoans as an emerging platform in nanotechnology of our topical bioactive principles. Here what I would like to tell you is, you all know about the phytochemicals. What are phytochemicals? They are all the active, pharmacologically active substances isolated from various sources, mainly plants. Okay, the plants are the vast sources for pharmacologically active medicines. Right or wrong? So we are isolating a pharmacological substance or therapeutic substance from the plant, and we are identifying its structure. We are comparing the structure with the standards that are available while confirming the pharmacological activity. So first we need to confirm the structure of the isolated compound by comparing with the standards that are available using various techniques: spectroscopical analysis, mass spectroscopic studies, IR spectroscopic studies. All these studies will help us to determine the structure of an unknown compound which is isolated from the natural source, isn't it? So before analyzing or before identifying the therapeutic activity of the drug, so we are determining the structure of the compound by using various advanced studies. So these methods will help us to determine the structure of the compound. By studying the structure of the compound, we are able to assess or we are able to compare the available drugs. We compare the isolated moiety with the available drugs, and we are determining the pharmacological activity of the drug, isn't it? So by performing various clinical studies, we are identifying the pharmacological activity of the isolated plant product which is having the therapeutic by various methods of pharmacological approach. Have you understood? So that we are formulating into various dosage forms, and those formulations are undergoing various clinical trials. By passing the clinical trials, they are available in the market, and they are used to treat various diseases. It is the basic pattern for isolation, formation of a new moiety as well as development of various formulations of the natural sources. So here we are concentrating on phytochemicals. As you all know that the active principles which are present in plants are not a single compound when it is present in its raw form. So there are n number of chemical substances will adhere and be associated with those compounds. We need to separate the individual compound to know its therapeutic action. Isn't it? So we need to separate and we need to isolate the pure compound. Then only we can easily identify the pharmacological activity with respect to the dose when we are using it. So here we should know about the various methods of extraction as well as isolation to isolate the pure compounds. You will be knowing about the isolation methods and extraction procedures to isolate the pure compounds from the natural sources. Okay, the pure compound which is in powder form or any other form, so which have the therapeutic potency or which have the therapeutic activity, they will be considered as the phytochemicals. What is phytochemical? The chemical structure which have the therapeutic potency obtained from plants. So they are called as phytochemicals. These phytochemicals are useful to treat various diseases, isn't it? At a particular dose in a suitable dosage. So first, I would like to give you a brief introduction about the phytochemicals. Coming to the phytochemicals. Phytochemicals. These phytochemicals are plant-based bioactive compounds produced by plants for their production through primary and secondary metabolism. They can also be derived from various sources such as food, whole grains, fruits, vegetables, and herbs. So basic by benefit of phytochemicals are they strengthen the immune system, they reduce the inflammation, they prevent the DNA damage, they also helps in DNA repair, they slow down the cancer cells for proliferation. Some of the diseases that are prevented by phytochemicals are cardiovascular diseases, obesity, neurodegenerative, diabetes, etc. These phytochemicals are also used to cure or to treat various metabolic, immunological, and neurological diseases. I am giving you few examples of various uses of these phytochemicals. So as you all know that the primary and secondary metabolites that are synthesized by the plants are utilized by the plants for its growth or as well as its survival, isn't it? Once the plant growth is stopped, what will happen? These secondary metabolites are going to produce the phytochemicals. Okay, it may produce the phytochemicals as well as the phytonutrients. Those compounds which have the therapeutic potency that we are considering as the phytochemicals, others will be treated as the phytonutrients. Example, vitamin C, etc. They will be considered as the phytonutrients. Next, the phytochemicals are compounds in plants. Phyto means plant in Greek. These substances are found in plant-based foods such as fruits, vegetables, whole grains, nuts, seeds, and legumes. They give plant the color, flavor, and aroma. Based on the presence of these phytochemicals or the chemicals that are present in the plant, they are only responsible for aroma as well as the color and flavor of the plants. Some of the plants which are rich in flavonoids, okay, their aroma is different from others. Okay, the aroma as well as the color or the flavor, they all depends on the amount and the nature of phytochemicals that are present in the plants. Phytochemicals can also exhibit their influence, including acting as substrates, cofactors, inhibitor of enzymatic reactions. So not only they are producing the therapeutic activity, they are also they can be used as substrates as well as cofactors and inhibitors of enzymes in various reactions. The antioxidant activity that have for phytochemicals is potential property. It has the ability to enhance the essential nutrients. Majority of the plant species which are rich in flowers, okay, they are rich in flavonoid because the phenolic compounds are rich in flower budding plants. Okay, these phenolic compounds have the antioxidant effect, okay, which is thereby produce the hepatoprotective activities. By demonstrating their chelating properties, they can get rid of undesirable constituent from shade. Okay, these phytochemicals, they also acting as chelating agents to remove the undesirable substances from shade. Some of the phytochemicals present in food that have the nutritional and energetic properties. That is why they are called as pharmaceutical compounds, because they are having nutritional as well as the energetic properties. The phytochemicals also have the ability to search the free radicals present in human tissue. Thereby, they are producing enhancing the immunity levels as well as they are enhancing the antioxidant properties of the tissue. The phytochemicals extracted from variety of plant species, those are being accomplished to treat different diseases. So as you all know that how we are extracting these phytochemicals by maceration or cold extraction, by using hot extraction, that is oxide extraction. Okay, the selection of Solvent is purely depending upon the nature of the compounds that can present in that particular species. Okay, we are choosing various n number of solvents to isolate or to extract as well as isolate the compounds. Thereby, we are determining the pharmacological activity of those compounds. So, okay, different plant species may have different phytochemicals which can be used to treat different diseases. Technically, though the term phytochemical is often refers especially to the compounds in plants, plant foods that are biologically active in terms of promoting health, but are not essential nutrients. Okay, as I said earlier, these phytochemicals, all the phytochemicals are not essential. Okay, some of them may also be used as nutrients. All the phytochemicals won't possess the therapeutic activity. So they may be used as nutrients. Example is folate as well as vitamin C, which are rich in food, and phytonutrients, rather than they can be treated as phytochemicals. Next, coming to phytosome. What is phytosome? Do you know the encapsulation methods where we are entrapping one substance in another, making into different formulations which are helpful to provide the better therapeutic activity and efficacy of the drugs. Do you know that? So here we are making the phytochemicals into phytosomes. We are converting the phytochemicals into phytosomes, which is done by encapsulation. We are using the encapsulation techniques to prepare phytosomes. We are converting the phytochemicals into phytosomes. So encapsulation is a process that entraps one substance within another substance and therefore produce the particles with the diameter of few mm to few mm, few nm to few mm. So we are entrapping the substance in another, thereby we are enhancing the particles. Encapsulation can change the liquids and other ingredients into powders, making them simpler to process and easier to use. As you all know that the therapeutic efficacy or the therapeutic activity of the drug is based on the amount that is used or the dose that we are fixing to produce that. The pure phytochemicals are most potent drugs. So while making it into a formulation, it is very difficult to make the formulation with the less amount of the substance. Okay, the phytochemicals are more potent and the supplement of phytochemical will be very less to treat any disease. You all know that for making a formulation, we require a particular volume of ingredients or particular volume or particular weight is required to make it into a formulation. So you all know that while making the tablets, we are making the granules by adding some binders as well as some natural gums or synthetic gums, producing the granules of different size, making it into a required volume or weight. Then we are making it into tablets, isn't it? Whereas the phytochemicals, which are most potent phytochemicals, when we are supplementing in 1 mg or 2 mg or 3 mg, making formulation will be very difficult. So we require to make it into encapsulated one so that we can enhance the particle size and we are making the liquids also into the powders to ease making of various dosages. It can also be used to improve freeze and thaw ability of sensitive ingredients by providing protection against moisture and cross contamination. Freeze and thaw ability, the condition explains whether the formulation is stable in various conditions or not. So we need to make a formulation in such a way that it should withstand all the conditions. It should withstand all the conditions and it should be stable at all the conditions. So encapsulation in food and dosage form used to mask the unfavorable odors of or taste. Phytosomes and liposomes are the examples of the encapsulated systems that are suitable in food and pharmacokinetic applications. Phytosomes is a technology developed by incorporating standardized plant extracts or water-soluble phytoconstituents into phospholipids to form complexes that have the ability to increase the extract's bioactivity. So here, what is phytosome technology? Here, it is developed incorporating the standard plant extract, which is a phytochemical, the standard plant extract or the water-soluble phytoconstituents into phospholipids. Say one of the type of lipids we are converting. The phospholipids are consisting of hydrophilic as well as hydrophobic moieties. So hydrophilic moiety as well as hydrophobic moieties are present in phospholipids. We are adding the phytochemical to the phospholipids to form the complexes that have the ability to increase the extraction bioavailability. Why we are adding this? To enhance the bioavailability of the extraction. It is added to the phospholipids. The phytosomes and liposomes are encapsulated systems with wider future applications in food industry. Therefore, in phytosome, phyto means plant, while some means a cell-like. What it means? A cell-like. Here, this is liposome and this is phytosome. This is phospholipid. So, which is having the hydrophilic moiety as well as the hydrophobic type. Okay, this is a drug. So, this is the complex how it develops during the process of encapsulation. The basic difference between the liposome and phytosome is the drug is within the phospholipid, whereas in liposomes, this is aqueous solution, this is hydrophilic head and hydrophobic tail. Have you seen this? So the drug will be in this area. If it is hydrophilic, it will be in aqueous phase. It is bilateral. So if it is hydrophobic, it will be there at hydrophobic phase. So it is not into the phospholipid. The drug is not into the phospholipid. That is the basic difference between liposome and phytosome. In phytosomes, that will be within the phospholipid. It makes the complex with the phospholipids by some chemical bonds. The hydrogen bond is most responsible for phytosomes. They are making the bond, they are adhering and they are present with the phospholipid. Head or tail of the phospholipid within the phospholipid. Phytosome technology produces a little cell like produces a little cell is able to better transit from hydrophilic environment into the lipid-friendly environment. And it enhances the bioactivity of phospholipids. This kind of phospholipids are consisting of phosphorus and hydrophilic and hydrophobic moieties used in phytosome production. Liposomes are smaller artificial vesicles of spherical shape that can be created from cholesterol and natural non-toxic phospholipids like this. Vesicle and what is meant by vesicle? A structure within or outside the cell consisting of liquid or cytoplasm enclosed by lipid bilayer. Okay. Liposomes also have been used in cases in which the lipophobic drug dissolves into liposomal bilayer. To overcome the low absorption of bioactive polyphenolic phytocompounds, nano-sized drug delivery systems can be utilized to enhance their preparation across the biological barriers by their unique physicochemical properties, increasing the bioavailability of the phytocompound. The major prospective of developing the liposome or phytosome is to enhance the bioavailability of the molecule or the phytochemical when it is entering into the systemic circulation or when it is reaching the target. So here the book how we are making it bioavailable, enhancing the bioavailability by enhancing the permeability by entering into the lipophilic as well as lipophobic structures. These phytosomes are helpful to provide the drug and to enhance the bioavailability of the drug. Now, the high surface area of the nanoparticles supports the effective delivery of active ingredients into skin in case of topical applications of the phytochemicals. So, majority of the phytochemicals utilized by public are in general they are nutritional supplements or cosmetic substances or some anti-inflammatory dosages, isn't it? So the basic thing what we need to study while making a formulation. So we should think how it is applicable topically. So what are the barriers for a pharmaceutical dosage form to make it into a topical application? So there are several barriers that we need to consider to make the formulation because it has to enter into hydrophilic and hydrophobic, lipophilic or lipophilic environment. So we should be very particular about this particular aspect. So the mechanism of polyphenolic chemicals incorporated into nanoparticles is by hydrogen bond or through hydrophobic interactions. The phytosomes are complex of phospholipids and natural active phytochemical bound to their structures, obtaining the reaction between the phospho-choline or any hydrophilic polar heads and the plant extracts in an appropriate sort. As I told you earlier, making a phytosome is very simple. So we are adding the phytochemical to phospholipid. Okay, we are adding the phytochemical to phospholipid in a suitable solvent. Where it is formed, it forms the hydrogen bond. The solvent that is used for making phytosome should be a protein. It should not donate or accept the hydrogen bonds because the phytochemical which is having the chemical structure on its own is composed of various hydrogen ions or protons, which makes the bond, the hydrogen bond with the hydrophilic substance or the moiety that is present in phospholipids. So here the solvent is inert and it is aprotic and it is not accepting the proton or it is not donating the proton because the hydrogen bonds that are formed within the phytosome is only due to the presence of hydrogens or protons in the phytochemical which is responsible for showing that particular therapeutic action. So that is why these phytosomes are most preferable when compared to liposomes because the drug is entrapped within the phospholipid and is reaching the targets. The phytosomes also called phytophospholipid complexes are the vesicular systems formed by interaction between the hydrophilic part of phospholipids and the phytoactive component, resulting in formation of hydrogen bonds between them. The structural difference between the liposomes and phytosomes is that the liposomes have the active ingredient inside the hydrophilic cavity or within the layer of membranes, where the phytosomes, those components are a part of the membrane, as I showed earlier. Okay, the drug is present within the hydrophilic moiety of the phytosome, whereas it is present in the bilayers of phospholipids in liposomes. The resulting complex helps these active ingredients to pass through the outer membrane of gastrointestinal cells and reaches their functional site. The absorption of these components is greatly increased and their bioavailability becomes much greater as compared to the original compound. So when the phytochemical is given directly, the bioavailability of the phytochemical in its phytosomal form is more. The bioavailability of drug in phytosomal form is more effective when compared to when it is given directly. So see the difference between liposome and phytosome. As I explained the earlier slide. So water soluble free drug. So majority of the drugs as are water soluble. So the free drug, that is, this is the drug. This is phosphatidylcholine, which is what is this? What is this? Phospholipid. Here, these are the hydrophilic heads. These are hydrophobic tails. So these hydrophobic tails are adhered together. Chusara there. So hydrophilic, hydrophobic, hydrophobic. So it is forming bilayer. Okay. So when it is passing through various systems, hydrophilic system or hydrophobic system in our body, so what will happen? The drug will not be absorbed by the fluids or any other hydrophilic and hydrophobic layers. So it has to reach the target site. It has to reach the target site with the help of this phospholipid bilayer. What we have done here? We have entrapped the hydrophilic drug into phospholipids and making it with a lipid. Whereas in phytosome, so as I said, the drug is hydrophilic in nature. So where it will be entrapped? This is the hydrophilic head and this is hydrophobic tail. The drug is entrapped in hydrophilic portion of the phospholipid, and this is phytosome and this is liposome. Bioavailability of drug is more in phytosome because it is present along with the phospholipids, whereas it is present within the bilayer that is produced by the phospholipids. Have you understood? This is the basic difference between liposome and phytosome. Difference between liposome and phytosome. See the phytosome. In phytosomes, the phosphatidylcholine and the plant components actually form a 1:1 or 2:1 molecular complex depending upon the substances or the complexes. So if you take 1 mole of phospholipid, or we have to take 1 mole of phytochemical or the active constituent, it is up to 1:2 proportion. If we take 1 mole of phospholipids, we can take 2 moles of biologically active substance. Wherein a liposome is formed by mixing water-soluble substance with phosphatidylcholine in definite ratio under specific conditions. Here it is not specified how much volume of the phospholipid has to be added, because it depends on the nature as well as the conditions that are required. So we can add whatever is required. Whereas the phytosomes involves chemical bonds, as I said earlier, they make the hydrophilic bonds, hydrogen bonds with the phospholipids. Whereas no chemical bond is formed in liposomes. The phospholipids molecules surrounding the water soluble substances. Phytosomes are much better absorbed than liposomes, showing better bioavailability. Whereas bioavailability of liposomes is less and compared to phytosomes. So next one is transferosomes. Transferosomes are vesicular carrier substance systems that are specially designed to have at least one inner aqueous component that is enclosed by lipid bilayer together with an edge aqueous. As you all know that surfactants, do you know the surfactant? Okay, the edge aqueous is nothing but the surfactant. Okay, the bilayer softly compound. It softens the bilayer. Examples are phenate, span 80 and sodium chloride. This aqueous pore surrounded by lipid bilayer makes ultra deformable vesicles having both self-optimizing and self-regulating capacity. Transferosomes are elastic in nature and can thereby deform and squeeze themselves as intact vesicles without a measurable loss through narrow pores in constrictions of the skin that are significantly smaller than the vesicle size. Transferosomes act as penetrating enhancers. They are acting as penetrating enhancers, felicitates drug molecule to penetrate across the stadium core. Okay, for topical applications. So the main barrier is stadium corneum, which is the outer layer of the skin. When these drugs are applied in various dosage forms as a liposome or phytosome or any other pharmaceutical dosage for any pharmaceutical dosage like cream or ointment, whatever it may be. So what will happen is it's a different situation because hydrophilic nature will be there and hydrophobic nature will be there. So these transferosomes are acting accordingly and the main target is to enter. So here we are using the alcoholic solvents for effective penetration into the layers of this. Whereas the liposomes or phytosomes, we have not used any alcoholic solvents. Whereas in transferosomes, we are using the alcoholic solvents, which will help them. We are also using the surfactants. The proportion may vary from transferosome to transferosome. We need to add or we need to use the alcoholic solvents as well as surfactants to enhance the penetration of these transferosomes. Okay, this is see how the drug will be entrapped in transferosome. So here it is there. Here drug is there. Here it is not entrapped. Here it is there. Here it is not entrapped. Have you seen this? Have you seen this? So it is flexible to penetrate into the layers of the skin and reaching the fabric. So this is about the transferosome. See, it is the hydrophobic phytochemical and this is hydrophilic phytochemical. You can observe the change in color of these two. This is phospholipid, as we all know. You have seen in earlier slides also. This is phospholipid. This is H activated or surfactant. H activated or surfactant. See, the transferable zones, this is hydrophobic component. Okay, these are the H activators. This is how the drug is managed in the bilayer of these phospholipids. With the help of these H activators, it is entering into the various barriers. Whereas liposome, the hydrophobic drug is entrapped between the bilayers of these phospholipids, which is different from phytosomes. Have you seen this? This is different from phytosomes. This is the basic difference among the liposome, transferosome, and phytosome. Where the bioavailability of the drug can be enhanced, definitely phytosomes. Coming to the next slide. Here we are differentiating the phytosome, liposome, and transferosome. Coming to the structure, the phytosomes, lipid bilayer vesicle composed of different type of phospholipids that can chemically bound to phytochemicals. What is the chemical bond that is present here? Hydrogen. Whereas in liposome, it is a lipid bilayer vesicles composed of wider range of lipids, including cationic, anionic, and neutral lipids. Okay. Whereas in transferosomes, lipid bilayer vesicles composed of phospholipids and H activator, that is surfactant, or the bites are ranging from 10 to 25%, low percent of ethanol and water as weight. So we require the polar or hydrophilic vehicle, that is nothing but the ethanol as well as water, the aqueous substance, which is acting as the vehicle. Here, coming to the encapsulation, the bioactive molecule, which is nothing but the phytochemical. The bioactive molecules are fixed by hydrogen bond to the polarity of the phospholipid. Choose polarity of the phospholipid. Whereas in liposome, the encapsulation is the active materials are incorporated into the aqueous core of the vesicles or in the lipid bilayer. In the lipid bilayer. Whereas in transferosomes, the active materials are incorporated into aqueous core of the vesicles or in the lipid bilayer membrane. And coming to the preparation of phytosomes, the phytosomes are prepared by selecting the suitable phospholipid and the phytochemical, the ratio of 1:1 or 2:1, and making the molecular complex that contains the chemical bond. Chemical bond is hydrogen. Whereas the liposomes, the lipid compositions mix alone, then the loaded materials added to the lipid thin film to form the complex with no chemical bond formation. Coming to the transferosomes are prepared by mixing an ethanolic phospholipid solution with an appropriate amount of H activator, that is nothing but surfactant, and buffering agent as hydrating molecule. So this is how the properties of phytosomes, liposomes, and transferosomes are varying one from another. Coming to the method of preparation, phytosomes are prepared by various methods. One is solvent evaporation method. Next one is mechanical dispersion method and salt out technique and lyophilization or freeze drying. In solvent evaporation method, it involves the integration of phytochemical constituents and phosphatidylcholine or phosphatidylethylamine or phosphatidylserine, that is PCPE and PS. There are the phospholipids that can be used to prepare the phytosomes in a flask containing the organic solvent in 1:2 molecule, 2 moles. This reaction mixture is kept at an optimum temperature, usually at 40 degrees centigrade, for a specific interval of 1 hour to achieve the maximum drug entrapment within the phytosomes form. Thin-film phytosomes are separated by under mesh, seeds and stored in desiccator for over. So because we need to dry the transferosomes, phytosomes, so we need to keep it in a desiccator for overnight. This is the solvent evaporation method that is used to prepare phytosomes. Next one is mechanical dispersion method. The lipids dissolved in organic solvents are brought in contact with aqueous phase containing the drug. The next, removal of organic solvent under reduced pressures results in the formation of phytophospholipid compounds. So this is how we are preparing the phytosomes using this mechanical dispersion method. In salt out technique, it is an important method of phytosome preparation that done by dissolving both the phosphatidylcholine and plant extract during a suitable organic solvent. Then an hexane was added until the extract phospholipid complex precipitation occurs. So we are making the precipitation of phytophospholipid complexes and we are using an hexane for precipitation. Okay. In this salt out technique. That's the reason why it is named as salt out technique. Whereas in lyophilization, it also like freeze drying. So in lyophilization technique, phytochemical will be dissolved in dimethyl sulfoxide. The resulting solution of 2.5% weight by volume was added to soy phosphatidylcholine dissolved in T-butyl alcohol of 1.5% weight by volume, followed by stirring for 3 hours on magnetic stirrer until the complex formation occurs. The complex was then dissolved and isolation will occur. This is called as lyophilization. So this is how lyophilization is a process in which water in the form of ice under low pressure is removed from a material by sublimation. So that is how we are preparing these phytosomes. Coming to the advantages and disadvantages of phytosomes. Advantages of phytosomes as compared to the conventional herbal formulation. You all know the regular herbal formulations. Okay, compared to the regular or conventional herbal formulation, phytosomes have the following advantages. It enhances the absorption of lipid insoluble polar phytoconstituents through oral as well as topical way, showing improved bioavailability, therefore extensively better therapeutic profile. So our main aim is to enhance the bioavailability as well as thereby enhancing the therapeutic activity of the drug, which is more in phytosomal form when compared to the regular conventional herbal formulations. When the absorption of active constant is enhanced, its quantity requirement is too reduced. So when the availability is there, we can reduce the dose. We can reduce the amount of dose that has to be supplemented. So which can be achieved by using these phytozoans. As the bioavailability is small, we can reduce the dose of the phytochemical also. Treat the particular this. Chemical bonds are created between the phospholipids, lipid molecules, and phytoconstant. Consequently, the phytozoans prove improved stability output. Here, the phytozoans are forming hydrogen bonds or other chemical bonds with the phytochemical, so thereby enhancing the stability of the formulation, isn't it? So when they are chemically bonded, they can improve the stability of the compound or the formulation. Additional nutritional profit of phospholipids. We are also providing the phospholipid along with the particular phytochemical in phytozoans when compared to the conventional herbal formulations. Considerable drug entrapment will be there in phytozoans. Application of phytoconstituents in form of phytozone improves their percutaneous absorption and act as functional cosmetics. Percutaneous absorption through skin. So what is the main aspect in preparation of cosmetics that are topically applicated? So it has to pass through various barriers that are there in skin. So which is more in phytozoans. Phytozoans process produces a little cell whereby the costly components of herbal extract are sheltered from damage by digestive secretions and gut bacteria. As I said you in the earlier slide, so we need to pass all the barriers to reach the target site. So these phytozoans will help. The phytozoans are used in handling of liver diseases including alcoholic, hepatic steatosis, that is fatty liver, drug-induced liver damage, and hepatitis. Okay. Phytozoans are useful in anti-inflammatory action, pharmaceuticals, and in beauty compositions. Phytozoans are used to treat acute and chronic liver diseases of toxic metabolic or infectious source of degenerative environment. Phytozoans are used in brain stimulant, immunomodulators. They can be used as immunomodulators, skin improvers, anti-wrinkle and anti-aging formulations. They are used as anti-cancer, antioxidant. They are used in hyperlipidemia, vein and skin disorders. They are used as cancer chemo preventive agents and used to care of benign prostatitis, hyperplasia. Benign prostate hyperplasia ఏంటంటే age associated prostate enlargement that cause urination disturbance. Okay. During to treat those particular conditions also we can use the phytozone formulations or we can convert them active principle into phytozones. They're also used to treat hypertension. The hepatoprotic effect of ginkgo biloba phytozone on carbon tetrachloride induced hepatotoxicity were investigated in rats. So they can be used as most potent hepatoprotective agents. It assures proper delivery of drug to the respective tissues. The nutrient safety of the herbal extract need not be compromised by conveying the herbal drug as means of phytozoans. These phytozoans have wide range of advantages when compared to regular herbal formulations. So we can use them to prepare anti-cancer agent, prepare antioxidant agent, prepare immunoboosters, to prepare skin preventive formulations. We can have n number of advantages with these phytozoans. entrapment efficacy is high, and moreover, predetermined, because drug itself is in conjugation with lipids in forming vehicles. So the drug is in combination with lipids in phytosomes. There is no problem in drug entrapment while formulating the phytosome, which are also superior to liposomes in skincare products. In skincare products. Coming to the disadvantages, even though we have various number of advantages with the phytosomes, the disadvantages are phytoconstituent is quickly eradicated or eliminated from phytosome. It is easily coming out from the phytosome, poorly absorbed due to their poor lipid solubility with hydrophobic substances. If the drug is hydrophobic in nature, poor absorption with it. When administered orally or topically, they limit their bioavailability. Phytosomes are unstable in nature, problem with stability. Phytoconstituents from phytosomes are rapidly eliminated. The duration of action is short, but advantage is the duration, even though it is having the less duration of action, it is more potent. The action is more potent. It has the short half-life. The possibility of phospholipid oxidation and hydrolysis reactions may occur in some cases. Leakage and fusion of encapsulation drug molecules also occurs with some formulations. These are few disadvantages of phytoconstituents. Coming to the pharmaceutical use of active constituents. We have the commercial products, the phytosomes are the commercial products. Coming to these commercial products, green tea phytosome, which has the active constituent epigallocatechin. This is the active constituent in green tea phytosome. The pharmaceutical use is antioxidant effect. It has the antioxidant effect, improved health and wellness. Grapeseed phytosome. The active principle is procyanidins, which have the therapeutic activities, natural antioxidant production. Whereas milk crystal phytosome, the active principle is cerebin. It shows antioxidant and stimulate the formation of new liver cells. Milk crystal phytosome. It improves the formation of liver cells, but it is not suggested to take directly or different doses. It enhances the liver cells and it is protecting liver and it will damage the liver also if it is consumed in excess. This most important thing while consuming this phytosome, we should take care of some precautions should be there. So we should take these medicine or advise these medicine based on the condition of the patient. Next, ginseng phytosome. The active principles are ginsenosides, provide adaptogenic function and resistance to stress, and it can be used as nutraceutical. Cerebin phytosome, that is nothing but milk crystal. The active principle is cerebin. It also have an activity of antioxidant and it also protects liver as well as skin. Next, centella phytosome. The constituents are terpenes, treat venous skin disorders. Ginseng phytosome. It is similar like that of ashwagandha, which is used in India, but ginseng is majorly from China. So these phytosomes are consisting of Panax ginseng, which is used as a tonic and skin tightener. So these are various uses of the active principles that are present in various phytosomes. Coming to the advantages of phytosomes in topical application. what is the main important aspect related to the topical application is they should cross various barriers of skin, isn't it? What is the composition of skin? What is the composition? It is the largest part of the body, and the surface of skin is about 1.5 to 2 meters in adults. Skin contains epidermis, dermis, and hypodermis. Such organ epidermis. It is a skin that of organ is the most important barrier for any topical application of various topical applicant formulations. So we should know about these aspects related to topical application. Status corneal. This is the dermis where we can see hair follicle, sweat gland. So this is hypodermis, a subcutaneous tissue where we can see arteries and vein. So this is how our skin appears. So we need to know about various aspects related to this. The epidermis is the most superficial layer of skin, and it varies in thickness in different parts of the body. The thickness of epidermis varies from part to part. There are no blood vessels or nerve endings in the epidermis. There are several layers of cells in the epidermis which extend from deepest generating layer to the surface stratum corneum. The cells on the surface are flat, thin, and non-nucleated. Dead cells are squamous. The squamous are the largest cells which can be present in normal urine samples also, in which the cytoplasm has been replaced by the fibrous protein keratin. It protects the skin from the harmful effects of sunlight. So the outer layer, the epidermis, protects the skin from harmful effects of sunlight. Next, coming to the dermis is tough and elastic. It is formed from the connective tissues, and the matrix containing collagen fibers bind water and use the skin layer. So this is about the dermis. Next one is hypodermis. It is the deepest layer consisting of adipose or fat that act as insulation and energy storage and fur and also cushioning. Coming to the advantage of phytosomes in topical applications, as you are aware of these advantages of phytosomes, we can see the same advantages here. That is, the potency of topical applicant can be enhanced. Phytosomes increases the skin absorption and bioavailability, and they induce the delivery of herbal active constituents or tissues. The high affinity of phytosomes to skin phospholipids intensifies its effectiveness compared to the conventional free compounds. So these are various advantages of phytosomes in topical application. So this is how these are various barriers for topical applicants to enter or cross the skin. So here, this is epidermis, this is dermis, and this is hypodermis. See the epidermal layer. It is stratus corneum, stratum, okay. ఈ లేయర్స్ అన్ని కూడా పాస్ అయినా ఎనీ డ్రగ్ వాట్ ఇట్ ఈస్ formulated in this topical combinations has to cross all these. These are all various barriers. So one to consider all these barriers while preparing the formulations which are useful as topical applicants. where the molecules can cross the SE via different pathways, which are either intracellular or intercellular. Intracellular penetration can be achieved via sweat glands, sebaceous glands, which secretes the liquids which are responsible for making protection of the skin from dryness or hair follicle, whereas the intracellular the transdermal permeability of topical products can be increased when the active ingredients are lipophilic and have low molecular weight. Okay, this is about the advantages. Advantage of phytosome. There is an important improvement in the absorption, so the dose frequency is reduced. When there is an improvement in the absorption, we can reduce the dose frequency. In preparations of phytosomes, the phospholipids are used, which also act as hepatoprotectant. Phospholipid contains much type of nutritional benefits. In skincare products, phytosomes is superior than liposomes. Phytosomes have high entrapment efficiency. They properly permeate to the skin. They are simple to manufacture in relation to other preparation, and there is no requirement of complication technique that is involved in making phytosomes. The disadvantages are phytoconstants are easily abolished from the phytosomes. There is stability problem in phytosomes. So evaluation of phytosomes. Phytosomes also be evaluated by using these techniques. One is visualization, and second one is particle size analysis and XRD analysis. Okay, these three techniques are useful to evaluate the phytosomes and compare it with the other formulations. So phytosome based bioactive phytochemical products available in market. As I said earlier, some of the phytosomes are available in market. Celibin from Silmarin, Marium, it is hepatoprotectant and antioxidant. Ginkgo, it is used as brain and vascular protection. Olive oil from European oil, it is used as anti-inflammatory, antioxidant, anti-lipidemic activities and cardiovascular. Centella, obtained from Centella asiatica, it is vein and skin used to treat vein and skin disorder. Green select, this is the phytosome of green tea, obtained from Camellia sinensis. It is acting as antioxidant. Antioxidant are nothing but anti-aging activity. So rutin is used to treat rheumatoid arthritis. Curcumin from Curcuma lama, it is used to treat hepatoprotective and used to treat the liver disorders. It can also have been used as anti-cancer curcumin. So this is how these phytosomes formulations are useful to treat various diseases. Phytosomes in clinical trial. See, the Silibin formulation. So it is used to treat prostate cancer. Now it is in phase 2 trials. The outcome will be, the study outcome is high blood concentration of Silibin. Okay, which will help protect the liver. Green tea extract. The main use of this green tea extract is obesity. Weight obesity. It is in phase 4 trial. So maintaining weight following weight loss. Have you understood? We should maintain the weight and during the weight loss. Grapeseed extract. Earlier stages of lung cancer can be prevented and this is in phase 2 trial. Okay. What it is doing? delaying the planned surgery more than 15 days. So it is showing some activity against the cancer cells. Vaccine, which is used in COVID-19, okay, it is in phase 3 under investigation. Philippine, okay, you can, it is used to treat the lungs, so it produces, it produces the liver cells. It is used to treat liver, produces the new liver cells. So it is under investigation. So this is how various phytosomal formulations are in clinical trials. Since 2007, the formulations of phytosomes started prepared by various pharma industries, and they are under clinical trials. So there are some assumption is that any formulation which is having the bioavailability with less adverse effects that can be used, but we should not consider this, and we should think about the antagonistic and synergistic effects of these phytosomes because they are with the phospholipids. So we should think the formulation, how these formulations are acting while used in various pharmaceutical dosage forms. What are the other substances that are present along with these phytochemicals? So if they also have some therapeutic activity, or if they act against the therapeutic activity, or if they are enhancing the synergic effect. So the dose, we need to add the dose that has to be supplemented. So what I would like to conclude is the targeting delivery of natural compounds and phytochemicals by using nanoparticles or nanocarrier can be significant benefit and have significant benefit, improving the bioavailability and biodistribution of the drug. So when it is absorbed properly, thereby it is enhances the bioavailability. When it is absorbed properly, from there it can be distributed properly. So the bioavailability and biodistribution will be enhanced using these entrap techniques with nanoparticles. Natural compounds and phytochemicals normally have low toxicity. The only issue concerns side effects induced by high doses of these natural compounds, which induce the problems with low patient confidence. The phytochemicals and natural products have a number of advantages, such as relative pharmacological safety when compared to the conventional formulations that are available. So phytosomes of anti-inflammatory phytochemicals exhibited anti-inflammatory activity above the standards. As I mentioned earlier slide, that is quasitine. It have exhibited more effect when compared to the other dosage. The phytosomes reduce the redness, itching, and inflammation of damages. The same were reported with antioxidant, hepatoprotective, and anti-proliferative effect against the human. The future perspectives of phytosomes. Phytochemicals have potential for various therapeutic and aesthetic topical applications. Due to their low absorption profile, nano-engineered drug delivery systems are used to enhance the penetration of bioactive polyphenolic phytocompounds across the biological barrier, thus increases their bioavailability. One of these novel nano platforms is phytosomes nanocarrier. The lipid composition and vascular nature of phytosomes enable them to penetrate skin layer in a more effective manner than the extract of phytochemicals alone. Have you identified this? The novel nano platforms use phytosomes, nanocarriers. The lipid composition and nanovascular nature of the phytosomes enables them to penetrate skin layer in more effective manner than the extract of phytochemicals alone. So phytosomes are similar to liposomes in terms of vesicular structure and have a similar stability and skin penetrating property. However, in phytosomes, the phospholipid interacts with the phytochemical via the formation of hydrogen bond between the phospholipids, polar head and polar functionalities of the bioactive compounds. Many phytochemicals have been formulated successfully as phytosomes, and there are likely to be other phytochemicals that can benefit from phytosomes forming. Future research might be considered using phytosomes with other phytochemicals or the incorporation of drug and phytochemicals the same nanovesicle to produce the synergy. There are any differences? Is it useful? Have you learned something? What is meant by phytosome? Any of us? What is meant by phytosome? Any of us? Encapsulation of phytochemical. Why? Chapter. Encapsulation of phytochemical. Phospholipid. So is it mandatory to keep a phospholipid to prepare phytosome? Phytosome. What's the basic difference between phytosome and liposome? Can anyone explain the structure of phospholipid? Can anyone explain the structure of phospholipid? See how if you are not satisfied also, I am happy because I am satisfied. So I have conveyed something and you have done something. So I thank all the organizers for providing me this opportunity. I once again thank the management. Do you have any questions?
Sir, I have doubts. Sir, phytochemicals are bio the good department as well as formulation, market model formulation. Phytochemicals convert to stability and the major problem. So how do we tackle the stability problem? So the necessary steps are these stability.
So when compared to the marketed formulations, there are no bonding in marketed formulations. We are not making any bonds. Here we are making a bond with phospholipids and as well as the phytochemicals, the hydrogen bonds. So thereby we are enhancing the stability. When the drug is available, biologically available for a certain time, when it is absorbed properly, the question of stability won't matter because what is our main aim? We need to transfer or we need to keep the active principle at the target site for treatment, not for a long time, but when it is consumed or when it is distributed and undergone ADME, again we need to supplement the other dose, isn't it? So here the stability is the problem, but to enhance the stability, these liposomes are converted into transferosomes. Thereby they are converted into phytosomes. So if we are enhancing the stability with respect to majority of these formulations, phytoformulations are topical. So majorly in general use phytosomes phytochemicals and skin protection gaani ledu antioxidant anti aging gaani ekkuva cosmetics gaani ledante supplements immune boosters gaani మనం యూస్ చేస్తాం. So thereby they are acting because if they are consisting of phenolic groups, which are nothing but flavonoids. So they have most effective antioxidant active because they are rich in phenol. So phenolic groups అంటే antioxidant వస్తుంది. Antioxidant వచ్చినట్టు definitely they may have the effect of protective. సో ఇక్కడ మనం చెప్తుంది ఏంటంటే when compared to the direct formulation, ఇక్కడ మనం stability అనేది direct formulation తో మనం కంపేర్ చేస్తే this is stable. అలా కాకుండా ఎలోపతిక్ మెడిసిన్ తోటో ఇంకొకటో మనం కంపేర్ చేసినప్పుడు దాని స్టెబిలిటీ అనేది తక్కువ ఉంటుంది. కానీ ఇప్పుడు డైరెక్ట్ గా ఫార్ములేషన్ ఏదో ఒక ఫార్ములేషన్ తీసుకుంది, ఇప్పుడు ప్లాంట్ ఎక్స్ట్రాక్ట్ తీసుకుంది, దాన్ని వేరే వాటిలో కలిపేసి మనం ఇచ్చేసాం. సో దాంతో పోలిస్తే వెన్ ఇట్ ఈస్ మేక్ ఇన్ ఫైటోస్, సో దీని స్టెబిలిటీ ఎక్కువ. దీని స్టెబిలిటీ ఎప్పటి వరకు ఎక్కువ? టు రీచ్ ద టార్గెట్ సైట్ రీచ్ అయ్యే వరకు వీ కెన్ మేక్ ఇట్ ఇట్ స్టేబుల్ వెన్ కంపేర్ టు డైరెక్ట్ గా మనం తీసుకునే ఫామ్. మనం డైరెక్ట్ ఓన్లీ మన ఎంజిఓ జిన్సింగ్ తీసుకున్నాం, ఇంకోటి ఏదో తీసుకున్నాం, దాన్ని తీసుకునే బదులు సిలిమారిక్ ఫైటోజోమ్ తీసుకుంటే దాని ఫార్ములేషన్ లో చేయడం మనం అవుతుందంటే సిలిమారిక్ ఫైటోజోమ్ స్టెబిలిటీ వస్తుంది. టార్గెట్ రీచ్ అయ్యి ప్రాపర్ గా డిస్ట్రిబ్యూట్ అయ్యి ఫాస్ఫోలిపిడ్స్ కూడా ఉంటాయి కాబట్టి, సో మన ఈ హైడ్రోఫిలిక్ హైడ్రోఫోబిక్ లేయర్స్ అనేది పాటుకుంటూ వెళ్ళాలి కాబట్టి, సో ఆ వాటి మధ్యన మనం ఆల్రెడీ మిక్స్ చేసి ఉంటుంది కాబట్టి ఆ టార్గెట్ అంటే స్టెబిలిటీ ఫార్ములేషన్ స్టెబిలిటీ చెప్పాలి అంటే వీ షుడ్ యాడ్ సమ్ additives to enhance the stability. That's the only option. Again, we need to add some additives. అలా కాకుండా ఇక్కడ మనం కంపారిజన్ చూస్తే ఇక్కడ స్టెబిలిటీతో మనం రెండు చెప్పుకుంటాం. ఈ స్టేబుల్ అని చెప్తున్నాం, స్టేబుల్ కాదు అని చెప్తున్నాం. స్టేబుల్ అంటే కంపేర్ టు ద యాక్టివిటీ ఇట్ స్టేబుల్. అంటే ఆ టార్గెట్ కి మనకు కావాల్సిన యాక్టివ్ ఇంగ్రీడియంట్ ని అది ఏమి డిస్టర్బ్ చేయకుండా, దాని కెమికల్ నేచర్ మారకుండా సేమ్ ఫంక్షనల్ గ్రూప్స్ ఉండేలాగా డైరెక్ట్ గా టార్గెట్ సైడ్ తీసుకెళ్తుంది బయట వస్తుంది. అదే ఫార్ములేషన్ స్టేబుల్ చేయాలి. ఇట్ డిపెండ్స్ అపాన్ ద నేచర్ ఆఫ్ ది యాక్టివ్ ప్రిన్సిపల్ అండ్ నేచర్ ఆఫ్ ది ఫార్ములేషన్. ఆయింట్మెంట్స్ స్టేబుల్ గా చేయాలి, క్రీమ్స్ స్టేబుల్ గా చేయాలి, లేదు సిరప్స్ అంటే ఆ స్టెబిలిటీ పారామీటర్స్. ఫార్ములేషన్ పారామీటర్స్ వేరు, డ్రగ్ పారామీటర్స్. ఎందుకంటే లిపోఫోబిక్ అనుకోండి, జనరల్ ఏంటి అన్ని కూడా లిపోఫోబిక్ ఉంటాయి. అంటే హైడ్రోఫిలిక్ డ్రగ్స్ కాబట్టి మనం యూజ్ చేసేది పోలార్ కాంపౌండ్స్ ఉంటాయి కాబట్టి టాపికల్ అప్లికేషన్స్ మనం. ఇది ఇప్పుడు మనకి ఎక్కడ ఇది వాటర్ ప్రూఫ్ లేదు. యాక్చువల్ మనం వాటర్ పడితే మళ్ళీ డ్రై అవుటర్ లేదు కదా. మనం టాపికల్ అప్లికేషన్ చేసినప్పుడు ఇప్పుడు మనం తీసుకునే మెడిసిన్ హైడ్రోఫిలిక్ నేచర్. సో ఇది ఏమవ్వాలి, ఇప్పుడు ఎస్సీ దాటుతాం, మనం డిఫరెంట్ లేయర్స్ చూపించాం కదా, అన్ని దాటుతాం. లోపల ఎక్కడో టార్గెట్ ఇన్ఫర్మేషన్ ఎక్కడ వస్తుంది మనకి, స్కిన్ లో రావాలి, లోపల వస్తుంది. ఇవన్నీ దాటుకుని వెళ్ళాలి. ఇవన్నీ దాటుకుని వెళ్ళే లోపు దీని స్టెబిలిటీ పోకూడదు. ఏది ఫైటోకెమికల్ స్టెబిలిటీ పోకూడదు. అందుకని చెప్పి మనం ఫైటోజోమ్ కింద కన్వర్ట్ చేసుకుని దాన్ని ట్రాప్ చేసాం. ఎంట్రాప్ చేసి ఈ లేయర్స్ దగ్గర ఎక్కడా కూడా ఈ లేయర్స్ పాస్ అవుతున్న స్ట్రెస్ వల్ల కానీ, ఈ హైడ్రోఫిలిక్ హైడ్రోఫోబిక్ రియాక్షన్స్ వల్ల కానీ, ఆ ఎన్వైరన్మెంట్ మారటం వల్ల వచ్చిన స్ట్రెస్ వల్ల ఎక్కడ కూడా ఆ కాంపౌండ్ పడకుండా, దాని కెమికల్ స్ట్రక్చర్ ఆల్ కాకుండా మన టార్గెట్ ని తీసుకెళ్ళి. సో దట్ we have achieved in phytosome. Formulation stability అనేది depends on the dosage form, suitable dosage form which you are going to select. అది మనం ఫుటిక్స్ లో చదువుతాం కదా, ఏమేమి వాడుకోవాలి, స్టెబిలిటీ ఎన్హాన్సర్స్. అది మళ్ళీ దీంతో ఇనర్ట్ గా ఉండాలి. ఇట్ షుడ్ నాట్ యాక్ట్ ఆన్ ఫాస్ఫోలిపిడ్స్ మీద కూడా అది ఇనర్ట్ గా ఉండాలి. అట్ ద సేమ్ టైం ఫైటోకెమికల్ మీద కూడా ఇనర్ట్ గా ఉండాలి. అట్లాంటి స్టెబిలిటీ ఎన్హాన్సర్స్ మనం యూజ్. సో అందుకనే కదా దాన్ని ఓవర్కమ్ చేయడం ట్రాన్స్ఫర్ ఏం చేసాం, ఆల్కహాలిక్ గ్రూప్స్ యూజ్ చేసి. ఇప్పుడు మనకు కొన్ని ప్రిపరేషన్స్ ఉంటాయి, ఆల్కహాల్ తోటే డిజాల్వ్ చేస్తారు. వాటర్ కంటే కూడా ఆల్కహాల్ తో డిజాల్వ్ చేయడం అంటే సో మనం ఈజీగా ఎవాపరేట్ అయిపోతుంది. కొన్ని పెయిన్ బాంబ్స్ ఉంటాయి, అక్కడ తీసుకునేది యాక్వర్స్ తీసుకోకుండా ఆల్కహాల్ తీసుకుంటాం. ఆల్కహాల్ బేస్ తీసుకున్నప్పుడు ఏమవుతుందంటే పెనట్రేషన్ ఫాస్ట్ గా ఉంటుంది, క్విక్ గా ఉంటుంది, కానీ అది లాంగ్ టర్మ్ కాదు. ఇమీడియట్ గా అక్కడికక్కడ అయిపోవడం కోసం చేస్తాం. స్టెబిలిటీ అనేది రెండు ఆస్పెక్ట్స్. ఇక్కడ మనం ఎన్హాన్స్ చేసిన స్టెబిలిటీ ఏంటి, స్టెబిలిటీ ఆఫ్ కెమికల్ సబ్స్టెన్స్. ఇట్ ఇస్ స్టేబుల్ ఐదర్ ఇన్ లియోఫిలిక్ ఆర్ లియోఫోబిక్ ఎన్వైరన్మెంట్. దాన్ని స్టేబుల్ గా టార్గెట్ చేయండి. ఫార్ములేషన్ స్టెబిలిటీ ఇస్ స్టేబుల్. అవి అండర్స్టూడ్. అదే మన క్రీమ్స్ ఉంటాయి అన్న, దాని స్టెబిలిటీ ఎన్హాన్స్ చేయాలంటే క్రీమ్స్ లో వాడే వేరే ఉంటుంది, సిరప్స్ లో వాడే వేరే ఉంటుంది. ఇక్కడ ఫైటోజోమ్ గా కన్వర్ట్ చేసేది పార్లెట్స్ కాదు, యాక్టివ్ ఇంగ్రీడియంట్ ఏదైతే ఉంటుందో దాన్ని ఫైటోజోమ్ గా కన్వర్ట్ చేస్తారు. ఫార్ములేషన్ ఈస్ డిఫరెంట్ ఫ్రమ్ ఫైటోజోమ్. ఈ ఫైటోజోమ్ ని సూటబుల్ ఫార్ములేషన్స్ మనం తీసుకెళ్తాం. ఇక్కడ ఫైటో కెమికల్ అని ఫైటోజోమ్ గా కన్వర్ట్ చేస్తాం. ఇన్స్టెడ్ ఆఫ్ నార్మల్ phytochemical, the phytozoans are more effective and more stable to cross various hydrophobic and hydrophobic. GIT లో chelating agents యాడ్ చేస్తే మనకి హార్మ్ఫుల్ లిక్విడ్స్ ఉన్నా కూడా ఆ టార్గెట్ రీచ్ అవుతుంది. ఫైటోజోమ్ ఫార్ములేషన్ వేరు, ఫైటోజోమ్ యాక్టివ్ ఇంగ్రీడియంట్ ఫైటోజోమ్. మీరు ఏదైతే ఇంగ్రీడియంట్ క్వాంటిటీ తీసుకుంటారు, పారాసిటమాల్ ప్రిపేర్ చేస్తారు. ఇప్పుడు 500 mg పారాసిటమాల్ తీసుకుంటారు, ఇక్కడ 500 mg ఫైటోజోమ్ తీసుకుంటాం. ఆ యాక్టివ్ ఫైటోజోమ్ మనం సేమ్ అది ఎలా తీసుకుంటాం అదే ఫైటో కెమికల్ ఫైటోజోమ్. ఫైటోజోమ్ ఇస్ నాట్ ఎ ఫార్ములేషన్.
Sir, we have a question from online participants. Are phytosomes similar to nutraceuticals? How about any safety regulations?
They are not similar to nutraceutical.
Yes.
We can prepare nutraceutical using phytozoa.
Yes. Fine. Sir. That's it.
Thank you, Dr. Narendra, for choosing this particular novel emerging technique, phytozoans, for today's talk. And firstly, I'd like to appreciate Narendra sir for coming here, especially despite of his health issues and also hectic schedule at his college. We should appreciate his interest in interacting the students physically. Actually, this is a hybrid mode FDP. We mentioned 2 times to sir whether it is online or offline, how you are willing for attend which session, like that I asked him. And sir want to interact especially with the students. So he is here today. Thank you so much, Narendra. And really, you emphasized very well about the novel techniques like phytosomes, liposomes, and even transferosomes. I think most of them don't know about transferosomes so far. So thank you so much for being with us today. And now I'd like to request the conveners of this session to felicitate sir.